A team of scientists at Harvard University are working towards creating a viable mammoth embryo within the next 2 years. Genetically engineered immune cells are saving the lives of cancer patients, with trials currently ongoing in China and soon the US. A recent breakthrough in gene editing technology is bringing headlines like these straight from science fiction into our new reality, and will undoubtedly alter what we perceive as normal forever. But first, some background. DNA is comprised of 4 paired-based nucleotides and makes up our genetic code. This code is organized into genes which carry instructions for the development of life. Change the code, and you change the organism carrying it. CRISPR is an ancient bacterial immune system used to defend against viruses. Bacteria have to defend themselves against so-called bacteriophages, or phages for short. Phages attack by injecting DNA into the bacterium and taking them over to use as factories. Bacteria which survive an attack can enable CRISPR. This involves saving a part of the phage DNA into its own genetic code in an archive called CRISPR. If the virus attacks again, the bacterium quickly makes an RNA copy of the saved DNA from the CRISPR archive. This so-called CRISPR RNA, plus another bacterial RNA called tracer RNA, is then used to guide a protein called Cas9, which scans the bacterium and acts as molecular scissors, cutting any matching viral DNA, making it useless and protecting the bacterium. Scientists adapted this system and turned it into a programmable tool. By combining both RNAs to create what's called a guide RNA, we create a simple 2-component tool. You give the Cas9 a copy of designed RNA and add it to the live cell to modify the DNA. The system acts like a guided missile, where the Cas9 is the missile and the RNA is the targeting system. This simple programming step makes it cheap and easy compared with previous gene editing technologies, so much so that anyone with access to a lab can carry out these experiments. Precise breaks in the DNA is a very efficient way of disabling genes. They do this by introducing small mutations which disrupt the DNA sequence. Repairing genes, however, is much more difficult. Scientists have to provide a DNA template which the host cell uses for repair. This carries a high risk for unintended mutations. The average human genome is over 3 billion base pairs long, and changes to a single base can be the difference between a healthy cell and one that's diseased, for example cancerous. So-called single nucleotide polymorphisms are difficult to repair using double-stranded breaks because of significant risks for mutation. We have already built a modified version of CRISPR that is made to change a single base letter without the associated dangers of inducing double-stranded breaks. The ability to restore these changes to normal could very well cure thousands of diseases forever. And as our understanding increases, we might be able to eliminate more diseases, including those associated with aging. Personalized medicines, where the treatment dies with the patient and is not passed on to future generations, are known as somatic gene therapy. Genetic changes made to future generations are known as germline gene therapy, and there is great controversy surrounding this subject regarding ethics and safety. It is important to understand that any germline therapy will begin aimed at removing deadly inherited disease, with no alternative treatment available. It is also important to understand that with currently available techniques, such as PGD and IVF, which involve selecting the healthiest embryos for implantation, there are few cases which actually call for germline therapy. However, in these cases, germline therapy is argued the correct decision, as condemning children to preventable suffering is unethical. These edits then open the door for further changes to future children, bringing the possibility of designer babies, children with engineered traits such as enhanced strength and intelligence. Things are picking up quickly, but before this future without aging and disease, filled with superhumans, is possible, a myriad of safety and ethical issues must first be dealt with. Safety issues include off-target effects, which can have dangerous unintended consequences, delivery mechanisms, and ensuring that all target cells are edited without eliciting an immune response from the patient. As these practical issues are quickly being resolved, we are pressed to address the ethical standpoint in determining the moral justification for gene editing, with questions such as, who decides who should be eligible for gene editing? Who decides the limits for these enhancements? Do the overall potential benefits outweigh the risks? And how do we ensure fair access for everyone? If we allocate these questions with just reason and experiments with due safety, then humans, the products of evolution, may one day become the masters of it.